The MAP kinase signaling pathway. The MAP kinase signaling pathway in the normal cell. The designation MAP kinase, abbreviated MAPK, originates from one of the first members of this protein family, the mitogen-activated protein kinase. The MAP kinase pathway begins with the binding of a ligand, for example EGF, to the extracellular portion of a membrane-bound receptor of the family of receptor tyrosine kinases, abbreviated RTKs. Ligand binding leads to the dimerization of two subunits of the receptor tyrosine kinase. At the inner side of the receptor, tyrosine kinase domains catalyze phosphorylation of itself and also of the other subunit. Next, the growth factor receptor-bound protein 2, GRAP2 for short, can bind to the phosphorylated RTK. The protein SOS, named after its homologue son of sevenless in Drosophila, is able to bind to the membrane-bound protein RAS. Inactive RAS is bound to the nucleotide guanosine diphosphate, GDP. SOS catalyzes the exchange of the RAS-bound GDP against GTP, guanosine triphosphate. This exchange leads to the activation of the RAS protein. In its active GTP-bound state, RAS is able to bind to several effector proteins. The kinase BRAF is one of the most important effectors of RAS. Active BRAF phosphorylates and activates the kinases MEK1 and 2, which in turn phosphorylate and activate the kinases ERK1 and 2. Finally, the kinase cascade leads to the activation of transcription factors of the AP1 family. The abbreviation AP1 stands for activator protein 1. The best-known AP1 proteins are the transcription factors Jun and Phos. After their activation, Jun and Phos move to the cell nucleus, where they form a heterodimer and bind to an AP1 motif of the DNA. This leads to the expression of many genes encoding, for example, growth factors, cyclins, and cytokines. As a